Hi, I'm Limeng, a PhD student from Plant Biology Department at the University of Georgia. Today, I'll present my poster, Phenotypic Spectrum, Quantifying Diversity of Root Architectures within Genotypes on the example of common bean. Root architecture is important for the uptake of water and nutrients. The divergent growth of roots at different classes, developmental patterns, and interactions between neighboring roots can result in extensive diversified root architectures, even within the same genotype. However, such root architecture diversity within genotypes is mostly ignored due to various reasons, of which a lack of computational tools is the most prominent. Here, we propose a shape descriptor, dx curve, to summarize the architecture of a single root. Using dx curves, we develop a computing pipeline to group similar architectures. Subsequently, we apply this pipeline to analyze a large image dataset that were collected in Arizona during the year 2015 and 2016. This dataset includes three common bean genotypes, DOR 364, SEQ7, and L88, in well-water and water-stressed environments. The pipeline includes 6 steps. First, we extract traits from root images using DIRT platform. Second, we use shape descriptor dx curve to describe root architecture as a whole. Dx curves are derivatives of the D curve that describe the accumulation of maximum width as a function of rooting depth. Therefore, dx curves describe the rate of root width changes by rooting depth. Third, we combine k-means plus clustering algorithm with Liddell algorithm to determine the optimal numbers of clusters. Then we simulate this process 1,000 times to get the best quality clustering result. Following that, we filter out clusters with less than 3 curves, shape outliers, and magnitude outliers. Step 5 shows one example of root architecture type after filtering. At last is the consensus match step. We identify similar root architectures across environments using the mean dx curve per identified architecture type in each experiment. We apply the French distance to quantify the similarity between mean dx curves. The blue areas in the matrix indicate high similarity. Because of the time and space limit, I will only talk about the result of one genotype, DOR 364. For this genotype, we identify 5 distinct root architecture types per treatment per year. If you look at the Fresh distance matrix, we can see 5 consensus architecture types across years and treatments, suggesting that these architecture types might be heritable. Second, we observe the ratio of root architecture types within the population varies among environments. So we can see in 2015, architecture type 5 decreased from 21% in water stress to 15% in well water. In 2016, architecture type 4 decreased from 25% in water stress to 18% in well water. The change of the ratios indicates that in certain environments, the population may prefer a specific architecture type. Interestingly, architecture 1 remains the dominant architecture type in 4 conditions. Third, we find the above-ground biomass does not differ significantly between root architecture types according to ANOVA test. So we hypothesize that the biomass homogeneity arises from the plant's strategy to optimize water and nutrients uptake on the population level. Based on our findings, we want to introduce the phenotypic spectrum as a new concept to describe the phenomena that a single genotype can display a finite number of distinct root architecture types in one environment. For future work, we will also investigate the effects of specific genetic or environment causes on the variation of the phenotypic spectrum. Thanks for your listening. If you have any questions, please feel free to email us.